Okay, welcome everyone. I'm seeing attendees start to trickle in for today's webinar. We have about 3 to 4 minutes until we'll get started. Welcome everyone. I think we'll give it maybe another minute or so to let more attendees come trickle in, and then we'll get started. Okay, well, it's 11, so on behalf of Biocytogen, I'd like to formally welcome everyone to today's webinar. We're very excited to introduce some of our antibody discovery experts that will provide some insights into the capabilities of our technology platform here at Biocytogen, so that we can ultimately support your ongoing research programs. Before we continue, I'll briefly introduce myself and the panelists. So I'm Jenna Frame. I currently manage scientific communications for the marketing team here at Biocytogen Boston. I'm joined by our speaker, Dr. Benny Young, and panelists who will answer any questions you may have after the presentation. So Dr. Zhang is the Associate Director of our Antibody Discovery team here in Boston, and he is also joined by our Scientific Director for Antibody Discovery, Dr. Hui, who is also supporting the antibody licensing efforts here at Biocytogen Boston. We're also joined by our Boston CEO, Dr. Ching-Kong Lin, who is also an expert in antibody drug discovery. He's joining us to round out the panel. So welcome to our panelists. And then finally, I'd like to formally introduce today's speaker, our Chief Scientific Officer, Dr. Benny Young, who has nearly 20 years of experience in immunology research and has been leading the antibody drug discovery team at our headquarters in Beijing since 2016. So I'd like to invite Benny to share his slides and begin the presentation. If you'd like to ask a question, the Q&A box is open and our panelists will address the questions afterward. So I'll hand over the floor now to Dr. Young.
Okay. Thanks, Jenna, for the introduction. Actually, I'm based in Beijing, so it's very late, but I'm very excited. So today's my presentation will focus on development of the best monoclonal antibody. And I will further discuss how we're going to use this platform to drive the research for bispecific antibodies and bispecific ADCs. So just mention a little bit background of Biocytogen's antibody research. So we started the antibody research drug discovery in 2016, and in the last several years, we have built, I think, very strong pipelines. So currently we have multiple assets in the clinical stage and the phase 2 or phase 1. This project are very special because we are using in vivo approach for the initial antibody screening. So basically, instead of doing the in vitro screening, so we started doing the in vivo screening and looking for the antibody hits that has the good efficacy and the good toxicity profiles. Then in the last 2 or 3 years, we started developing a new class of antibodies, which are bispecific antibodies and the bispecific ADCs, using our newly invented technologies, and which I will be explaining in more details. So the first thing is to share a bit information on the discovery and the research on the fully human antibodies. So I think 4 years ago, we did an analysis looking at all the approved drugs that time by FDA and the EUA. So in that 10 years, we found that there's a very clear trend that most therapeutic antibodies are fully human antibody, or at least humanized antibodies. This is make sense because you can minimize the in vivo anti-drug effect. So these antibodies are less immunogenic once you inject into the people. But there's different ways you can generate this antibody. One is the in vivo approach, the other is the in vitro approach. Actually, both approaches are successful. It has led to the approval of several drugs. But you can see very clearly the field like the in vivo transgenic mice to generate fully human antibodies. This is because usually the in vivo selection is more powerful, and also the quality of the antibody binders are better in terms of the CMC developability. But whatever approach you take, when you're doing the antibody research, you have to think about 2 things. The first thing is, once you have a approved antibody, you ask what's the diversity of these clones. When we mention diversity, we talk about the sequence diversity and also the epitope diversity. So the more diverse, it's more likely you will find good antibodies. The second thing is the quality of these antibodies. So usually people measures the quality by looking at the binding strengths. So we usually call the affinity measurement. But the other thing is you also need to pay attention is the CMC developability. So we always want to lower the risk at a later stage. So if the antibody have good physical chemical properties, and these antibody are more likely to success in the later stage of development. But I will focus on the in vivo approach using the transgenic mice to generate the fully human antibodies. In the past 20 years, actually, the field has developed multiple mouse models for this purpose. And actually, these mice can be divided to 2 generations. The first generation actually is using the knockout approach, and then put the human antibody genes in using a transgenic method. This including the famous mouse used by BMS, used by Amgen. This mice is successful, but they also have a problem because the knockout transgenic approach does not drive very potent immune response, and usually the mice are the immune response is generated in these mice inferior to the wild type animals. But later, Regeneron came up with a new technology, which is in situ replacement. Basically, we are putting the antibody variable regions in the endogenous locus of mouse antibody genes. But at the same time, they keep the constant region intact. So these mice usually develop better antibody response and also better B cell development. And other examples are KY mouse or Triani. At Biocytogen, in the last, we spent 6 years developed a new mouse model called RenMap. This mouse is also considered as second generation fully human antibody transgenic mouse. We also did in situ replacement using a novel technology. So this allows us to replace the entire variable region with the human counterparts. This has never been achieved by any other companies. So that's why I think this is by design, this is considered as the most sim mouse, have the most more like what we see the antibody diversity in humans. So how this mouse is generated? So this is actually using a technology called Super-C, stands for size unlimited chromosome engineering technology. But basically, we can put any fragment at any desired locus in the genome. So for example, for the heavy chain, what we want to do is to replace the 2.6 megabase variable regions of the mouse locus with the counterpart from a human, which is 1.0 megabase. So this consists of more than hundreds variable genes, diversity genes, and join genes. So these are the VDJ genes from the human locus. So what we did here is we replaced from the very beginning to the very end the entire locus. So we keep the constant region of the mouse antibody locus intact. So this come up with this structure. And we did the same thing for the kappa chain. And the kappa chain is very unique because it consists of 2 clusters of genes, the distal cluster and the proximal clusters. In the past, people always want to, because the difficulty of gene replacement, they usually only replace the partial of the kappa chain locus. But since our technology is not limited by size, so we can replace the full fragment. But in theory, we can replace any size. So this is not a barrier for us. So we cross these 2 mice together, then we got a mouse containing the human variable region of the heavy chain and also the kappa chain. So this is how the RenMap mouse is generated. So with this mouse, the antibody discovery is much easier because we only need to inject human antigen into the RenMap mouse. So we can get induce the antibody response from this mice. Then we clone the sequence and put the variable region onto the human FC expressing vectors. Then we really get a fully human antibody. But this it takes a very only a couple of weeks, and then you get a genuine fully human antibody. But otherwise, if you don't do that this way, the traditional approach is to immunize wild type mice. Then you get a fully mouse antibody. Then you can generate the chimeric antibody. Then it takes a long time and a lot of expertise to doing the humanization. And eventually, you will get a humanized antibody, but it's much tedious and also there's a lot of uncertainty involved in this process. So the RenMap really offers a good advantage of generating high quality, fully human antibody. So when I mean high quality, I will give you an example. This is actually a project we did to looking for the neutralizing antibodies against COVID-19. So basically, what we want to do here is to find the cocktail antibody, which is like a 2 or 3 antibody. They can simultaneously bind to the viral protein, and they block the interaction of the virus and the host protein. So at that time, we immunized the RenMap mouse with the RBD region from spike protein, and we get a bunch of high affinity binders. And more than 10 of these binders can block the interaction of the RBD and the spike and the human AC2. And we did in vitro characterization looking at their binding epitope. And we can divide these clones into 4 different bins. basically some of these antibodies can bind, their binding epitope are not overlapping, means they can simultaneously bind to the spike protein. So this actually, we can further confirm this data using the cryo-EM. So we resolved the structure of 3 different clones binding to the receptor binding domain. And you can see this actually is exactly confirmed what we predict from our epitope binding assay. So further, we showed if we combine two antibody or 3 antibody, basically, and test against an array of viral mutant, we can find with single individual antibody, they cannot prevent the viral escape. This you can see are very many red boxes. But if we using 2 or 3 antibodies together, especially if we do have combined all 3 different antibodies, they say it can completely eliminate the viral escape from the neutralizing antibody. So this actually is a way to prove how the RenMap can generate high quality therapeutic antibodies. But the viral protein is one thing, but usually we're dealing with host protein, which is like we immunize mouse and human. We really need to consider about one thing is how to break the tolerance, because many proteins are highly conserved between mouse and human. So here we did a research comparing the homology of 150 targets that we are interested in. And then we found that actually most of our targets have a conservative over 50%. This is really considered as a barrier for immunologists. So to make it more clear, I show some example here. So these are the proteins have different homology between the mouse and human. For example, CD73, it's 90% conserved between mouse and human. So we colored the amino acid that are identical between mouse and human with green color. And you can see most of the surface of this protein is colored with green, which means they are, for the immune system, these are considered as a blind spot. So usually when you immunize a mouse with a human CD73, you are likely to generate a few antibodies against those yellow residue. But for the green epitopes, it's not possible because of the tolerance mechanism. So I think this is actually one way we need to address, one thing we need to address for generating diverse antibody response. How we can do that? Actually, we come up with this way is to knock out the specific target on the RenMap background, and then we immunize a human protein, and we will not have any tolerance issue. This actually shown in different projects. Actually, I compared and I summarized some data for 6 different projects. These are their homology of between mouse and human are typical for like between 53 to 94%. It's very typical candidates. So if we using regular RenMap mouse do immunization, and then we go through the high throughput screening with the Beacon single cell system, so we can get for example for target A, we can only get 100 binder. But if we knock out that particular genes, now we can get a 600 binder, 6-fold higher number of hits from this screening. And it's same thing for all the other targets we studied. So this really tells us if we knock out a particular gene, usually we'll get a higher antibody titer, which I did not show here, but also we can get a higher number of binders on the Beacon system. So this really drive us to coming to start this large campaign called Project Integrin. In this Project Integrin, we are looking for developing the high quality antibody for every single gene we are interested in. So, but we will always consider first knock out that particular any gene on the RenMap background to facilitate the antibody discovery. For example, if you do not do knockout for this protein, you're only likely to get those yellow antibodies against those non-conserved epitopes. But if you knock out a particular gene, then you can also get not only yellow antibody, but also get those red antibody against those conserved regions. And all these red antibodies recognize conserved epitopes across mouse and human. So this really also add another additional advantage for antibody screening, because once you have antibody recognized mouse target, you can directly do screening in wild type animal. But otherwise, you have to do the screening using target humanized animals. So this really not only means you can get a high number of antibody binders, but also these binders you can do is much easier for screening. But so the screening process get much straightforward, which will accelerate the translation from the preclinical study to clinical study. So I will give you one example here is the project for CD45. This is one of the project we in this Project Integram campaign. So for this one, basically the first thing we did is to knock out CD45 on the RenLite background. This take about a half year, but I think it is really worth. So then we do the conventional antibody immunization, then do the single cell discovery for any antibodies. So usually we can get more than like 200 binders, potent binders on the beacon system. Then we clone the sequence of these binders, and at Biocytogen, we always prefer to do the in vivo screening first, then followed by all the in vitro characterization. And then I think so. This is basically one of the development process for one of the project. So this is, I think, will really change the diagram of the antibody research, because in the past, the whole field spent 40 years and only leads to the approval of more than 100 drugs, and this only covers about 64 genes. But actually, there's many genes have not been thoroughly explored clinically or preclinically. But we think there's actually, we did a study using the database. We think there's more than 1,400 genes that can be targeted by actually several genes targets that can be studied using antibody therapies. And I think we using this Project Integram, which will generate a larger number of hits against many targets. This will really help the antibody researchers in the field and for exploring or testing novel targets. So as I just laid a little background on the fully human antibody, but now I will spend more time to discuss bispecific antibodies and the bispecific ADCs. So while we talk about the bispecific antibody, I think this is actually become a really now a hot areas. And but as many people have impression, so there's more than 100 different format of bispecific antibodies, and every company have its unique platforms. But how we can how they are different from each other. But I think regardless of the format that people are using, but there's several things we need to keep in mind to really understand the bispecific antibodies. The first thing is important is the valence of the antibody, which means how many binding arms on the antibody against the specific target. And usually we're also looking at the appearance of the antibody, whether they have FC version, part or no FC, whether they are symmetric or non-symmetric, whether how they are the different part of the binding domain are connected with each other, like a head-to-head or tail-to-tail configurations. And but also for antibody, the epitope always is important issues. And most also for bispecific antibody, and epitope and affinity, and even the balance of affinity between the two targets is really the key for the successful development of bispecific drugs. But regardlessly, I think all the antibody, major form of bispecific antibody can be categorized into two different classes. The first one is the top one, like we call 1+1, and also we have the 2+2. The difference between this is the top class of antibodies. They look like conventional monoclonal antibodies, but they have the two arms have different binding specificities. Basically, the left arm binding to one target, and the right arm binding to a different target. The prototype for this class of bispecific antibodies is a cross-map, but later many other companies developing new technology to achieve the same function. The second class of bispecific antibodies is actually much easier to construct because you only need to engraft a binding domain of a second antibody onto an existing monoclonal antibody. and you come up with this, like usually it's 2+2 configurations. I would say both formats are useful, depends on your purposes. But today I will focus on the top class of bispecific molecules, because they provide a uniqueness, beauty of this class of molecules, which is the selectivity of the drugs. So give you some example of this class of drugs. Basically, people have using this mechanism to construct many different drugs. Like for example, you can this is a molecule. It's called biparatopic antibody. So both arms bind to the HER2, but they bind on the different epitopes. So one is against domain 2 and the other against domain 4, which is actually considered is more potent than conventional monoclonal antibody. And also you have antibody, bispecific antibody bind to 2 different tumor-associated antigen on the cancer cells, and this drug has been developed by Johnson & Johnson and was already approved. But there's many other ways you can you can think about how this molecule can be used, and even including co-engage 2 different cell types. One arm bind to the CD3 on the T cells, the other arm bind to the tumor-associated antigen on the cancer cells, and which drives the killing of these cancer cells by immune cells. So this antibody, I think the beauty, as I mentioned about these antibodies, they really because the monovalent from each arm. This antibody, when you combine 2 different selectivity into the one single molecule, this molecule usually provides better selectivity over the conventional monoclonal antibody. So we're not going to use the cross-map approach, but so instead we develop a common light chain solution for generating bispecific antibody at a very high throughput. So this model is based on the RenMap. In this mouse, we decided to replace the carbon chain with a single rearranged sequence. This sequence is from 3-11. So this is a productive sequence, and we replaced using this sequence to replace the entire variable region of the carbon chain. So in this mouse, all the B cells will have the almost identical light chain sequences. So the only thing different from different B cell generated in this mice is the sequence from the heavy chain, which is still the product from the VDJ recombination and the somatic happen mutation. So basically, using this approach, we can solve the mismatch issue of the heavy light chains. So so using this mouse, basically you can immunize 2 different antigens, like you generate the red antibody against target A and the green antibody against target B. Then you assemble bispecific antibodies. So light chain makes sense because both arms using both target using the same light chain sequence. So you don't need to worry about that. So the only thing we need to resolve is how to have correct pairing of the heavy chain. But the field already had a lot of experience in dealing with that. So basically, there's many different options for that. So I will show you how we can do that. So bispecific antibody can be easily assembled just by using some very mature technologies. But otherwise, this cannot be achieved if you're just using a wild type animal. So basically, which will give you a mixture of antibodies which cannot be separated easily. So as I said, so we already considered about 2 things for evaluating the new models. The first thing is the diversity. Yes, the second thing is the quality. So when we look at the diversity, I show the data from 2 different projects. So each project we immunize the RenLite and mice. So we get more than 1,000 binders. So we look at the sequence diversity, and basically we look at which B genes, which D genes, and which J genes are used. But basically, here we show that the different J genes is shown using different colors. But basically, you can see for both targets, these binders we got from the immunized mice actually are using different combination of V genes, D genes, and J genes. So the sequence is very diverse. But also, when we look in the affinity of these binders, actually they are actually in the starting from the, this is measured using the SPR, SPR system. So the binding affinity is starting from the 10 to the minus 8 to up to 10 to the minus 12. So basically, these are in the typical range of monoclonal antibodies. So which is good for the drug development. So the next thing is that how we can efficiently pair the two different arms into a single molecule. So I use the data from HER2 as an example. So basically HER2 has 4 different domains, and basically you can target 2 different domains using the binder have different specificity. So we pick the clones bind to the domain 2 and also clones from domain 4. Then, as I said, the only thing you need to worry is the heavy chain mispairing. So we using the different technology, which has already been published. So including the knobbing to whole from the genetic, and the Maris solution, and the Zencor solution, and the ZamiWork solution. We found that just after when we co-express this heavy chain light chain in a single cell, and just after single step protein A affinity purification, we can get very pure band on the SDS PGGR. The only one have you can see like a half molecule is the one from technology using the ZamiWorks. So basically, actually, I think this data shows the RenLite sequences are very friendly to all the heavy chain pairing solutions. So then, if you have already recognized this, you will find the generation of bispecific antibodies is very straightforward. So basically, we're always starting with immunization of the common light chain RenLite mice. If possible, we are always first knock out the particular target on the mice background. Then we are using our high throughput beacon system to doing the antibody screening. And usually we get 100 binders. Then we are first do the epitope binding first, and to find the clones have different binding epitope and also different like binding affinity. And then when we want to construct a bispecific antibody between target and target B, we just need to co-express the two heavy chain and a single light chain in the same cells, and just after one step purification using the supernatant, we'll get a pure bispecific antibody product. Then this can go to the downstream analysis, including in vitro functional assays, in vivo efficacy study, and also we also always pay a lot of attention on the physical chemical analysis to show this molecule is really convenient for the latest development. So now I will give you some examples to show how we're gonna using these tools to generate novel drugs. The first thing I talk about today is to generate a Treg targeting bispecific antibody. So Treg is a really good target for cancer immunotherapy, and there's many targets highly expressed by the Treg cells, but always we found these targets all have a certain level of expression on other T cells, especially the active effector T cells in the tumor. So we want to use the bispecific approach to generate a molecule that can recognize 2 different targets on the Treg cells. So this will minimize the binding on the activated T cells. So this is the rationale of this project. So we first show if this can be done with a high quality protein can be generated in this project. So this is just show the data of the the purity and the stability of this our candidate. So this is shown after just single step SEC purification. You can see homogeneous thrashing, which is like very decent. So this thrashing is stable at the storage condition. So we store the molecule at a high temperature, 40 degrees for up to 4 weeks. We analyze this solution using the SEC to look in the purity, and we use the HIC to look in the hydrophobicity of this molecule. And you see over weeks, this protein remains, the profile of this protein is very constant. And this can also be shown in the ICIF to show the charge heterogeneity of the molecule. And even after 4 weeks storage, we don't see the profile of this molecule change. So we also compare the in vivo potency of these molecules. So we compare combining with the 1+1 format of this bispecific antibody, compare with the parental monoclonal antibody, which are bivalent. So this study is done using humanized mouse models, which we humanized both targets on this mouse background. Then we give the drug and did a head-to-head comparison. And you can see in this is the first experiment, which show the bispecific antibody. It is more potent in inhibited tumor growth in these models. And we further want to repeat this data. We can see those dependent response. So confirm what we found. But also not only for the targeting two different cell receptor on a Treg cells, we think the bispecific technology can also be applied to the antibody drug conjugate because they can provide a higher specificity toward the tumor cells. So we show that here all the ADC approved in the last several years. Actually, you can see more drugs in the ADC field have been approved, especially in the last 3 or 4 years. And this target covers all the blood malignancy, but also the other solid tumors. And many different payload linker technology have been used, but still, I think the MMA still dominate and the approved drugs. So actually, in our internal project, we still using MMA because our technology is to not on the payload linker side, but on the antibody side. We want to generate a better antibody to drive for this to provide more better drug candidates. So the question here, actually, using the bispecific platform is to provide better tumor cell selectivity, and then leads to the effective tumor cell killings without affecting normal cells. This is actually what I use a cartoon to show what I mean here. So usually tumor cells expressing different markers compared to normal cells. So they usually expressing not only more than one tumor-associated antigens, and the normal cell always express low tumor-associated antigen, even if they express. But if we can using a bispecific molecule can target two different tumor-associated antigens, they say this antibody can bind to the tumor cell much better than healthy cell, which express only one receptors. But also for the bispecific ADCs, I think the conjugation of two different receptors also have another beauty, is that they drive the internalization of the receptor, which is a requirement for the payload link to work inside these cells. So, but there are not many bispecific ADCs in the clinical trial. So far, there's only 2 we can find in the database. One of the molecule is bispecific ADCs targeting MUC1 and the EGFR. This is sponsored by EMD Serrano. Basically, this molecule MUC1 and EGFR are highly expressed, and also they co-express on many different tumor cells, but their expression level in the normal tissue is very low. So this is why the rationale behind this molecule. But the other one is actually it's a biparatopic ADCs. So this molecule recognized two different epitope on HER2, and also I think this will, but it is conjugated with a payload linker to drive the killing of these HER2 expressing cells. So for us, I think when we design and constructing the bispecific ADCs, we think the three things are important for the product. The first thing is how you're going to select the target. The second thing is what's the relevant functional assay for the selection. So for us, I think the answer is very simple. The endocytosis is the key. The third thing is the stability. Since we are studying bispecifics and also these molecules need to be conjugated in vitro. So the stability or convenience is really the one important thing to consider. So first, how do you gonna select the target? So we analyzed more the database. This is public database, and more than 1,000 human cancer cell line. We look at the tumor associated antigen expression. We find that so here, that's just actually HER2, ERBB2, and TROP2 are co-expressed by many different human cancer cell lines. But if you look at the individual cell cancer type, you will have the same impression. But we really want to ask whether they are indeed co-expressed by the same cells. So we're using an internal PDX bank. So, and we did a single cell RNA-seq analysis. So this actually, the data I show here is a gastric PDX samples. So this sample had been passed in mice. So we did a single text genomics-based single cell analysis. And then we analyzed this data, and we can clearly see this population, the population 0 is coming from human, but all the other actually are coming from the immunodeficient mice. So we further see the expression of HER2 and the TROP2. We found indeed you find the expression in those human-derived cells, but not in the mouse-derived cells. Importantly, HER2 and TROP2 are co-expressed by the same cells, which is clearly shown in the panel C. So then we decided to actually develop high quality binders for these tumor-associated antigens. Our approach is still very straightforward. So we start with the immunization of RenLite or RenLite target knockout mice. Then we do the beacon screening, and we first validate the binding on these cells using the FACS analysis. And we choose more than 100 strong binders, and we then do all the functional relevant assays, including endocytosis. Then we check the cross-species activities, and we measure the affinity by the SPR technology. And then we choose some top clones, then we assemble them into the bispecific ADC format. Then when we do all the downstream in vivo and in vitro characterizations. So our list of tumor-associated antigen actually is more than 200 targets. I think these are if you are interested in, we can share this information. So I will give you this is a real case. So now this asset is in the CMC development. So this is a bispecific ADC is targeting HER2 and TROP2, the YH012. So here, as I said, we still want we are trying to optimize the antibody part, but not so we still in we are not designing new payload linker. So we're still using the most successful linker payload, which is MMAE. So here we did all the comparison of this novel bispecific ADCs with all different controls, including parental monoclonal antibodies HER2 TROP2 or monovalent antibodies of HER2 TROP2. For these monovalent antibodies, basically the one arm is still binding to HER2, but the other arm we using we replace with an irrelevant antigen. So we're comparing this HER2 TROP2 ADC with all the controls. So what we predict is this. So this molecule will bind to tumor cells expressing both targets, but the normal cells express HER2 and TROP2 at a very low level. So the antibody will ignore these cells. This way, the antibody will provide very high specificity toward those cancer cells. So still show the stability and the developability of these molecules. So we need the SEC, HIC, and the ICIF to analyze this molecule, conjugate molecule, stored at different conditions, like high temperature at 40 degrees for 1 week, at the multiple rounds of freeze thaw conditions to see whether the molecule remains stable. And at the low pH conditions, we find that at all the tested condition, this molecule have very good performance. And when we do the transfection, just by putting heavy chain, light chain into same CHO cells, we can get a very decent expression level over 0.4 milligram per ml. And just up to 1 step of purification, you can get a single peak resolved by SEC. And this molecule has the typical conjugation profile. So we use MMAE to conjugate this payload linker onto the cysteine residue. So this is actually unconjugated molecules. This is the 2 payload, 4 payload, 6 payload, and 8 payload. So if you are familiar with MMAE technology, you will see this is very typical to a conventional monoclonal antibody conjugated with MAE. So this makes sense because the bispecific antibody we conjugate really have very similar profile comparing to the monospecific antibodies. So we also did some in vitro and in vivo study, including the endocytosis analysis. So we in the InQ side to comparing the endocytosis efficiency on a tumor cell line, which is positive for HER2 and TROP2. So here we're comparing, we find YH012 is endocytosed more efficiently comparing to the parental monoclonal antibody or monovalent antibody. It is also more efficient endocytosis comparing to the TROP2 benchmark and the HER2 benchmark. So here we also plot the data in a different way, showing that on the right panel. In the in vivo study, we also chose a lung cancer cell line expressing HER2 and TROP2 confirmed by FACS analysis. So in this assay, so we implanted the tumor cell into the immunodeficient mice, and we just give 2 shots of the other drugs. So we still see in the YH012 is most potently inhibited the tumor growth. All the other controls, including parental monoclonal antibody or monovalent antibody, or the TROP2 benchmark, HER2 benchmark, they all work, but none of them can compete with YH012. I think this is another way to show the beauty of this molecule. So actually, I think now it's I want to just quickly summarize what I mentioned in this talk. I think we provide a very powerful platform which can robustly generate high quality antibodies using the RenMap or RenLite. And for RenLite, especially, I think you are convinced that knockout the target on this background really can make the antibody discovery much easier. And we can easily generate many binders for different target. And then we can using the high super approach to assemble a bispecific antibody for further modifications, including we can conjugate this antibody with different linker payload. So we have tried so far we have tried the conjugation on the cysteine residue, but I think this will be work for our different conjugation technology, because this molecule they behave just like monoclonal antibody, which is very friendly. So this antibody really provides a higher specificity toward the desired population, which will increase the safety of these drugs in the trials. So I think this is the last thing. So I want to summarize my talk. So I think at Biocytogen, we provide novel mouse models for antibody research, including the RenMap for the fully human antibody, the RenLite for the common light chain solution. So currently, we are also developing the third mouse model in this family, which is called RenNano. So this is a fully human antibody generate transgenic mouse, but in this mouse it will generate single domain antibody, or we call heavy chain only antibody. So we have some preliminary data. As far as I can tell, I think this is the best heavy chain only antibody I think I have ever seen. So I think it would be very exciting to fully launch these models to the field. But so far, actually, I think this we are building a project, what we call RenLite hits, which hits stands for hyperimmune target specific. Basically, it means we would like to knock a particular target on the RenLite background to facilitate the antibody research. But basically, we are doing a lot of knockout project on the RenMap and the RenLite, and so far, I think we have knocked out more than 980 target on this mice background. Many of these mice we already get the F0 homozygous mice and F1 and F2, and also have some mice we already started immunization and or already have the started antibody screening, and more than 180 target we already have high quality antibody hits. And I think if you are interested in doing antibody and you are looking for existing like available clones, then maybe this will be very useful. I think if then the last slide is about the BD strategy. I'm not going to talk about that, but I think we have been collaborating with many different companies, and basically we are our BD strategy is very flexible and open. So we can work with different ways with licensing or co-development, or just licensing the technology. I think if you are interested in, I think you can talk to our BD personnel, and I think we have. So I think that's it. Thanks very much for your attention.
Thank you so much, Benny, for that very comprehensive overview. So it looks like we're out of time for today. I think if there's any questions that we weren't able to answer, we will reach out to you and clarify as needed. And you can also contact us by email at info@biocytogen.com, or head to our websites, biocytogen.com or renmab.com, to get more information about our platforms or the mouse technology platforms. We'd love to hear from you and answer any questions through those methods. And thank you so much, Dr. Young, for that wonderful presentation and for joining us late at night.
Thank you, everybody. Thank you, Jenna.
Thank you so much.
Thank you, Benny. Thank you, everyone.
Thank you.